Okay, everyone.
So my name is Laura Burnham, and I'm the project manager of CHAMPS. And today I would like to introduce Dr. Lori Feldman-Winter, the professor of pediatrics at Cooper Medical School of Rowan University. And she's also our CHAMPS physician lead for the CHAMPS project. And today she's going to be talking about or presenting on why not just one bottle, the art and science of exclusive breastfeeding.
Thanks, Laura. So welcome, everyone. I just want to remind you again, if you can place your phones on mute. If you're using the computer and you see a little telephone by your name, then an easy way to do it is just to click on that phone icon and you'll get a little X. If you're just a call-in user and you can't identify yourself by name, then you can use *6 to mute. If you press hold, sometimes we'll get your hospital's advertisement, and that's entertaining but probably distracting for the rest of us. So please don't just push the hold button on your phone, because that could funnel us into some music. So without further ado, I'm really excited to meet everyone by phone today and really begin the dialogue about what's needed for step 6, which is the support for exclusive breastfeeding and really decreasing the rate of supplementation. And the first part of understanding and then making the changes necessary to implement step 6 in doing that is to first ask the question why, and that's what this webinar today hopefully will do. Will be a discussion. Hopefully we'll have some questions and pushback from all of you. You know, I really want you to challenge me. I'm going to present some of the evidence that we know about why it's important to support exclusive breastfeeding, why it's so important not to give not even one bottle or one drop or 1 teaspoon of formula. I have no disclosures other than, as Laura said, I am the physician lead for the CHAMPS project, and I think I already covered the objectives. I want to really delve into some of the more microscopic reasons for why the need for exclusive breastfeeding is now known much more than it has been in the past. And so I'm going to describe a little bit about what happens in the development of the newborn's immune system and how that, in fact, protects the baby not only in the short term but in the long term. And as this slide illustrates, there are a number of different health outcomes that are associated with breastfeeding. This is a summary from the ARC review that was conducted in 2007 looking at pediatric health outcomes. And the ones that I've circled, 1 through 6, are the diseases that are linked not just to any breastfeeding, but really to the exclusivity of breastfeeding. And so what I bolded is exclusive breastfeeding, EBF, exclusive breastfeeding. So things like ear infections or atopic dermatitis, lower respiratory tract infections, the serious ones that lead to hospitalizations, asthma, obesity, and type 1 diabetes, at the time of this ARC review had evidence that there was a link not only between breastfeeding and preventing those diseases, but the need, the importance of exclusive breastfeeding. So it wasn't just any breastfeeding, but really the requirement of exclusive breastfeeding to help offset some of these diseases. Interestingly, some of the things in the latter half of this list, 7 through 10, are some of the more long-term health outcomes associated with breastfeeding. And at the time of this review, we didn't have a tremendous amount of evidence to support the link between breastfeeding exclusivity and long-term benefits. But in a recent study that was just published, looking at leukemia as an outcome measure, and I think a lot of people don't really think about childhood cancer as being a disease outcome that could be related to infant feeding, but in fact, leukemia and lymphoma are 2 cancers that are in fact linked to infant feeding method. And in this study from Australia, they examined cases of acute lymphocytic leukemia. They also looked at brain tumors in children that were under 15 years of age, and then they selected control subjects from random digit dialing. And they examined a number of cases. In the case of the brain tumors, there didn't seem to be an effect of infant feeding, but there was clearly an effect of any breastfeeding and the reduction of risk for acute lymphocytic leukemia. But I think what's most dramatic in this recent study is they looked at the age of formula introduction, and it was really just the formula introduction within the first couple of days of life that had the strongest link, about a 50% increased chance with an odds ratio of 1.57, causing an increased risk of acute lymphocytic leukemia. It's not just the milk itself, but we think a lot of things about breastfeeding, and really breastfeeding at the breast confers disease protection. And in this study that was done from the CDC's infant feeding study, there was evidence that it's not just about the properties of human milk, but the method of feeding at the breast that was most likely to prevent how often an infant would overfeed in the latter half of the first year of life. So each one of these percentages is the percent chance that a baby would overfeed in the latter part of the first half of life. And in their multivariate analysis, they found that the babies that were bottle-fed intensively, so babies bottle-fed either formula or breast milk, were 2 times more likely to overfeed. But in a dose-response method, those babies that were bottle-fed exclusive breast milk were more protected than those babies that were bottle-fed formula. So it's really a combination of breastfeeding plus the properties of human milk that really leads to a lot of the disease protection that we see. This is some of the evidence that we at the American Academy of Pediatrics use to support the argument and finally the consensus that exclusive breastfeeding should be supported for 6 months, no longer 4 to 6 months or about 6 months, but really for 6 months. In this study, they examined about 3,500 infants throughout the first year of life to determine the influence of exclusive breastfeeding for 6 months versus 4 to 6 months versus less than 4 months compared to those babies who were never breastfed on outcomes of upper respiratory tract infection, lower respiratory tract infection, and gastroenteritis. And this is the slide that illustrates the effect of just in the babies in the first 6 months of life, where in all cases there was a significant decrease in the risk of all 3 of these diseases, only for the category of exclusive breastfeeding for 6 months, not less than that period of time. So it's important to continue exclusive breastfeeding. Once the cohort was examined in the latter part of the first year of life, the effect on gastroenteritis, interestingly, disappeared. So the babies were just as likely to be protected if they had been exclusively breastfed for less of a period of time. But the outcomes of upper and lower respiratory tract infection remain. So the disease protection, again, outlasted the period of the exclusive breastfeeding, since this was a period after the introduction of complementary foods. And in addition, in a more recent study looking at ear, sinus, and throat infections, and these were outcome measures that weren't even included in the ARC report, what was clear in this study, again, is that the exclusivity of breastfeeding, so breastfeeding duration for more than 6 months, but without formula introduction, was really the only category that led to protection from ear, sinus, and throat infections beyond infancy. And they examined this cohort, again, through the infant feeding survey. in children up to age 6 years of age. So there was a significant decline in the risk of these 3 diseases in children up to 6 years. So we know that there's evidence of epidemiologic evidence of immune modulation because we know that the diseases that are affected by exclusive breastfeeding really occur well beyond the period of exclusive breastfeeding. But how that happens is quite complex, and it's really just now that we're beginning to shed light on how human milk confers the protective effects that last beyond infancy. And it's been speculated that the immunological factors in breast milk influence the development of the infant's immune system such that the pathogenesis of disease later in life are actually decreased. And now we know that the relationship is really one which includes not just human milk, but with the proper colonization of what we used to call commensal bacteria, and now we understand it's really much more of a mutualistic relationship between the proper colonization of gut bacteria in the setting of exclusive human milk, and that the bacteria support the development of the immune system. Similarly, the proper immune function of the baby's gut hosts the proper immune microbes. So it's really more than just a commensal relationship. It's really a symbiotic or mutualistic relationship between the gut of the newborn and the proper intestinal microbial organisms and human milk that support this setting. And I'm going to delve a little bit more into this in the next few slides. We also have epidemiologic evidence that infants that are exclusively human milk fed are less likely to have allergies, and really any formula in the first year of life leads to an increased prevalence of atopy or allergies. But really, allergies are from a combination of factors, and there are a number of non-modifiable factors that lead to allergies. But among the modifiable risk factors, infant feeding comprises the largest category. So we know it's something that we can do differently that will help reduce the prevalence of asthma and allergies as well as atopic dermatitis. So how does it work? Well, to understand how it works, we have to really understand how human milk influences the development of the infant's immune system. And when we think about the immune system, we think about 2 big categories of how our immune system functions. One is in the category B down below in adaptive defenses. And I will tell you, 20 years ago, this is really all we knew about the immune system, that through adaptive defenses, our bodies made B cells, which made antibodies, and T cells, which led to the T cell or cell-mediated types of immune protection, things that would prevent against viruses or parasites. And that was really all we knew. But really recently, over the last 20 years or so, we've begun to at least scratch the surface in understanding what's probably an even more complex part of our immune system through innate defenses. Some are easy to understand, such as the skin or the mucous membranes producing mucin that provide protective barriers that are nonspecific. But what's really more intriguing is how all of these other factors within innate immunity and the innate defense system, through T cell regulation and this beautiful cell that I've now added to the slide, the dendrite, actually communicates between innate defenses and the adaptive immune system. And so these 2 arms of our immune system are not quite as distinct as the slide would initially illustrate, but really a complex interaction between not so specific immune factors and the specific parts of our immune system. And hopefully I'll be able to illustrate some of this as we go on. What's essential is that the gut has to be properly colonized to do this work. And in fact, the newborn gut must be exposed to a whole series of bacteria shortly after birth. We used to think that the gut was actually sterile in utero, and now there's some newer research leading to the thought that maybe the gut has already some of the commensal or more mutualistic bacteria even in the fetal gut. But we know that exposure to the flora that surrounds the mom's vaginal canal is important to begin the process of proper immunological responses as well as colonization. And so if the newborn's not exposed, if the baby's developed via C-section, if the baby gets antibiotics, if there's ways that normal microbial environments are somehow altered, then not only does the immune system not develop properly, but that leads to allergy and other autoimmune-related problems. So shortly after birth, what happens is really a series of very closely regulated processes. In utero, at about 19 weeks, the newborn's intestinal immune system is pretty much structurally present, really waiting for major inductive processes to occur right after delivery. And what happens is that, as I've illustrated here, through a big globule of colostrum coming down, through mom's colostrum, which contains the commensal bacteria as well as secretory IgA and TGF-beta, there are major inductive processes that occur at the layer of the gut-associated lymph tissue. The gut-associated lymph tissue is comprised of Peyer's patches, mesenteric lymph nodes. There's a surface epithelial barrier, as I referred to earlier, as this mucin or nonspecific protective surface. And then the lamina propria have abundant lymphocytes and macrophages and dendritic cells, like that dendritic cell that I showed you just before. And they're all sitting there waiting for these major inductive processes that occur through colostrum. And in fact, without the colostrum and the commensal bacteria and secretory IgA, what fails to occur is the organization and development of that sort of amorphous structure that sits in the middle of this slide, this isolated lymphoid follicle, or ILF. That isolated lymphoid follicle with that core of B cells that is depicted by the sort of oblong-looking B pink structure, surrounded by the shell of T cells, is really the origins of the entire immune system for that baby. And it's through the colostrum that that isolated lymphoid follicle actually develops, becomes properly organized spatially and also genetically through what's happening in the transcription of the genes of those cells, and then also in terms of where those cells then go after they leave the gut-associated lymphoid tissue to circulate throughout the rest of the body. And I'll show you what happens in terms of the development of the lymphatic system a little bit later. So really during this process of bacterial colonization of the newborn gut, many bacteria are brought into immediate contact with the baby's gut. And again, they're being transferred from the mother's own gut through the vaginal canal, as well as some through the mother's colostrum. And human milk actually protects the newborn from becoming antigenically sensitized to some of the potentially pathogenic bacteria that are present in the mom's vaginal canal. They have to first exist and first be present on the surface of the intraepithelial cell, as is illustrated here. And then the colostrum permits these harmful pathogens to be removed without an inflammatory response. And I'm going to discuss how that occurs. And finally, through the secretory IgA, and in particular, there's anti-idiotypic secretory IgA that the newborn essentially becomes immunized. So what does this mean? Well, an antibody has what's called an idiotype, and that idiotype has a structure that is in the mirror image of the shape of antigens, and there can be a zillion different antigens that exist in the world. And what mom's milk in the production of her milk does is actually take the antigens in mom's environment, and through the construct of secretory IgA in the manufacturing of human milk, makes the antibody IgA against those different antigens in the environment, but also makes the mirror image of the antibody. And so the mirror image of the antibody is essentially a safe antigen. So it's really a prepackaged antigen that's less harmful than the sort of wild-type antigen that then gets transferred to the baby, and in cooperation with bioactive cytokines that are also present in mother's milk, permit the baby to actually undergo antibody production that the baby wouldn't otherwise undergo. And so in a sense, the baby is literally becoming immunized through mother's milk, through these anti-idiotypic antibodies, and inducing immune memory that leads to both short and long-term protection. And so protection from diseases, but protection from other things that are affected by the immune system. And we believe now, we understand a lot more in the oncology literature how closely linked oncology and cancers are to defects in the immune system. So where did the bacteria come in? Well, the enteric bacteria, these happy, friendly, symbiotic bifidobacteria, actually participate in organizing these immune cells, the B cells, T cells, and macrophages and dendritic cells at the level of the intraepithelial cell. And this is a beautiful electron micrograph shot by Borchelli and Walker from Harvard, showing how those bacteria are actually stuck to the cells and participating in regulation of antigen transport and in pathways of recognition to host specific bacteria and other factors that should be hosted, and in other cases really get rid of factors that shouldn't be there. And in the process of getting rid of pathogens that shouldn't be there, it's done in a way that's anti-inflammatory, doesn't require an inflammatory response. And that's so important because we think that it's the improper inflammation that exists without the presence of these commensal bacteria that actually leads to damage of the newborn's intestine and leads to things such as necrotizing enterocolitis. So how are these bacteria supported? Well, the bacteria are actually supported through nourishment that's also manufactured in mother's milk. Human milk oligosaccharides is the primary nutrient of the commensal bacteria. And the majority of the carbohydrate that exists in human milk is lactose, and that's for nutritive purposes of the baby. But a huge part of the non-nutritive part of carbohydrates in mother's milk is in the form of these human milk oligosaccharides. Those oligosaccharides serve as the substrate for the fuel for these microorganisms, and the specific human milk oligosaccharides that nourish the commensal-friendly bacteria are the ones that are present in mother's milk with a wide repertoire, mother-to-mother difference in specific nuances of how those human milk oligosaccharides are actually developed. And so how do they work? Well, they work by either in the first cell of this slide serving as a ligand to support the connection between the surface carbohydrates of the cell and the bacteria. So in this case, the human milk oligosaccharides would be hosting. They serve as nutrients, but they can also serve as immune protective factors in that they might bind receptor sites or block receptors from allowing certain bacteria that shouldn't be there to actually be rid from the baby's intestine. So again, those more pathogenic bacteria can be blocked through mother's human milk oligosaccharides. And this is, again, another electron micrograph just showing that the sugars on normal surface cells, in this case in the throat, in this particular case, permit the bacterial adhesion. This is not a good situation because pneumococcus is the thing that is being adhered to, and in this case that would lead to infection. But in other cases, oligosaccharides might thwart the attempt for bacteria to enter the cell, again by binding to those receptors, not allowing the bacteria to bind. So in many ways, the human milk oligosaccharides themselves participate in immune protection for the baby. So the probiotic bacteria, or the commensal bacteria, the bifidobacteria, essentially needs an invitation to stay in the baby's gut, and it's invited through complex mechanisms, but also through this prebiotic or carbohydrate that serves both to allow them to adhere as well as serving as a nutrient for the bacteria to grow. So in the natural sense, the normal bacteria are hosted without having to use an inflammatory response to rid the pathogenic bacteria. And this is illustrated again through a study looking at the pH of the stool in exclusively breastfed babies compared to formula-fed babies, and then they studied a fermented formula to see if that even made a difference. And throughout the first 4 months, it was clear that the breastfed babies had the lowest pH, an indicator that these commensal bacteria were, in fact, fermenting the carbohydrate. But oligosaccharides are, while they might be necessary, are not sufficient to properly colonize the infant's intestine. Another factor that's, again, another indication for the need for exclusivity comes into play, and this is in the toll-like receptors. Toll-like receptors are these receptors that exist primarily on dendritic cells, that popular cell that I showed you early on. And those toll-like receptors are necessary to help bind specific bacteria, and in some cases bind them in ways that lead to cell death or apoptosis, which sort of in a graphic way blows up the cell and gets rid of the pathogen, and in other ways host the bacteria to be present and living in a symbiotic manner. And you need certain toll-like receptors to be present at sometimes and others to not be present. So certain toll-like receptors like TLR1 and 2 are the ones that are responsible for binding gram-positive bacteria, whereas TLR4 are responsible for binding the gram-negative lipopolysaccharide producing more harmful pathogenic bacteria. And it's in studies that were done looking at the transcription of these different toll-like receptors, namely the difference between the transcription of TLR2 and 3 and TLR4 and 5, that shed some light on how exclusive breastfeeding really mattered. And I don't want you to focus too heavily on all these really complicated graphs, But essentially, the message is that human milk, and not infant formula, and not any derivation of it, because they really tried to manufacture different types of formula that would mimic human milk. But it was really just in these hash bars where it was exclusive human milk that potentiated an upregulation of TLR4 and 5 responses. So the toll-like receptors that would rid the lipopolysaccharide bacteria. And interestingly, there was a negative effect on human milk, so a negative signaling effect of human milk causing decreased transcription of the toll-like receptors 2 and 3, again, permitting the hosting of the bifidobacteria. And so specific toll-like receptors were necessary for proper colonization. TLR4, again, being the one that was upregulated, necessary to rid the pathogenic bacteria, doing so through this T-cell apoptosis, as illustrated on this slide, and then the downregulation of TLR1 and 2, allowing the gram-positive bifidobacteria to be hosted. So exclusive human milk is necessary for proper colonization, and as those experiments illustrate, just one bottle or just any interference with the exclusivity leads to a change in this very specific temporal relationship between how the toll-like receptor transcription is tightly regulated over the first few days. And they actually looked at, in their experiments, different transcripts, and they saw really on an hour-by-hour, day-by-day basis changes in the setting of exclusive human milk, or really colostrum, that allowed for differences in how the signaling occurred in making the toll-like receptors. So going back to allergy and how human milk affects allergic responses, we know that developing an allergy or atopy and ultimately asthma is an effect of the Th2 cells, the T helper cell 2s, which leads to an increase in the eosinophil production and also an increase in the B cells that will eventually produce IgE antibodies, and those IgE are the things that allow the mast cells to release their granules and lead to all the effects of itching and allergy. And newborns are naturally skewed to producing more Th2 responses than Th1 responses. So newborns are naturally hyperimmune, if you will. And so it's a requirement for human babies to be exposed to colostrum so that these Th2 responses, these hyperallergic responses, are really tempered by more tolerance that occurs through Th1 responses. It's really Th1 responses requiring, as I've illustrated here, requiring the commensal bacteria and secretory IgA to drive tolerogenic responses, tolerance to different antigens without being inflammatory and allowing for less autoimmunity later in life. And so it's really all about balance. You can't have too much of one thing or you end up with disease. So you can't have too much Th2 or you end up with allergy, but at the same time you don't want too little Th2 responses or you might get infection with things like parasites, which is really the first thing that newborns' Th2 systems respond to. Same thing with Th1 responses. You don't want too much Th1 responses or you'll have too much cellular apoptosis, but you can't have too little Th1 responses or you may have too little tolerance. And that's really simplifying it by looking at just those two arms of the T-cell immune system, because there's a number of different categories of T-cells that also come into play that are tightly regulated. So as I've illustrated here, it's really all about balance. And as I talked about earlier, it's really those T cells that then circulate from the earliest time of inductive processes throughout the baby's body. And what's been shown is that breastfed babies have a thymus gland that's twice the size of the formula-fed baby, and that these T cells that initially develop in the gut are sent to the thymus gland and really function for the entire life of that baby. And I think that this slide is probably, for me, some of the most convincing evidence that exclusivity really makes a difference. What this slide shows is differences in the composition of white blood cells or in immune cells in babies that are 6 months of age. And in the study, they looked at differences between babies that are exclusively breastfed, babies that were formula-fed, and they actually studied another formula that was higher in protein, thinking that some proteins might affect immune composition in a more positive way. And what they found in both the study formulas as well as the standard infant formulas, really the composition of immune cells at 6 months was remarkably similar. And it was really only the exclusively breastfed babies that had distinctly different patterns of immune cells at 6 months. The formula-fed babies had more T cells that were skewed toward naive T cells. They had a decrease in the natural killer cells, decrease in the cytotoxic lymphocytes and B cells, and a slow recruitment of the T cells with those effector functions that are so important for that innate immunity. And interestingly, the patterns were essentially the same as the infant formula-fed babies if the exclusive breastfed babies were introduced to infant formula at 4 to 6 months. So really the exclusivity for that full 6 months was necessary for these proper compositions of immune cells at 6 months. And as I alluded to, there's something about human milk that actually leads to cell triggering, to the signaling of the nucleus to transcribe specific gene patterns. And this is a study that looked at the cell transcription for master genes that are actually useful in angiogenesis and wound repair. And what they found, again, is distinct differences in the breastfed babies in gene expression in these master genes that are responsible for angiogenesis. So another piece of evidence to say, yes, human milk doesn't only produce the cellular elements and the cytokines that lead to proper immune function and the commensal bacteria, along with secretory IgA and TGF-beta, but also participates in cell signaling that allows for different transcripts to either permit different genes from being permit different genes to be transcribed or prevent, in some cases, certain genes from being transcribed. Additionally, Ward and colleagues have studied the human milk metagenome. We know that there are a number of genes that exist in our guts, in newborn guts, in response to just the intestinal microbiome. In fact, just due to our normal host intestinal microbiome, there's about 100 times more genes that are present in our bodies than are from our actual human genome. And this has drawn a lot of attention for researchers looking at the functions of these genes. And in studies looking at the metagenome of human milk and comparing the genome that exists in mother's milk to the genes found from the stools of the mom and the breastfed baby and also the formula-fed baby, they found that in the mother's milk, there's a metagenome that actually contributes to proper colonization and immune modulatory effects of the baby's gut, because there are genes that have open reading frames, which means that they're open to be transcribed, and they participate in these stimulatory effects, or in some cases suppressive effects, of the DNA that is, again, present in the mother's human milk that lead to effects in the infant's intraepithelial cells. There are these dinucleotides that are present in response, that are present because of bacterial DNA. There are what's called CPGs, or two nucleotide strands that actually participate in some of the effects of immune modulation in the baby's intraepithelial cells. And so what ends up happening in disease protection goes beyond the actual genetic structure of the infant's own genes and the genes that might be in the intestinal microbiome. But in addition, there are factors that are related to infant feeding that lead to different patterns in what we call epigenetics. And the epigenetics really basically is a way to describe whether or not the genes that are there are actually going to be transcribed or not. This is an effect on the genes that leads to either up transcription or decreased transcription that leads to differences in patterns in how the baby's cells are going to either manufacture or not manufacture certain proteins. And we know that there are differences in the byproducts of the bifidobacteria in the baby's gut that leads to the promotion of DNA methylation, which can, again, change the way genes are transcribed compared to infant formula. And this, in fact, is presumably one mechanism that leads to changes in metabolism. So as I talked about earlier, the effect of human milk on obesity prevention is thought to be due to the properties of human milk as well as the process of breastfeeding, and this may be one of the mechanisms related to the properties of human milk, and it may be in the effects on epigenetics. So in summary, the neonatal microbiome participates in the baby's development of proper immune cells, and there should be this balance between B and T cells for a properly functioning immune system so that the baby develops tolerance when tolerance should be present, immunity when immunity should be present, and then getting rid of infections or potentially pathogenic microbial organisms in a way that's anti-inflammatory, that's not causing damage to the host gut cells. In addition, the baby has to develop proper immunity in a way that's not hyperallergenic or leading to autoimmune diseases. The signals that occur in the baby's intraepithelial cells and in dendritic cells are not just at the surface, but actually are upstream and actually occur at the level of the nucleus. So the normal neonatal microbiome is not just a way to properly develop the infant's immune system, but really an indicator that the infant's immune system is properly functioning. And really any formula feeding and potentially through these other mechanisms, through antibiotics or C-section, there can be disruption. There are a number of different research questions that are now focused on the neonatal microbiome, and it's exciting because the NIH is funding a lot of new projects that are looking at the microbiome of pregnant women, looking at the microbiome in terms of prevention of inflammatory bowel disease, a condition that has really exponentially increased over the past several decades, and also looking at the way in which the neonatal microbiome might participate in chronic diseases, things such as type 2 diabetes. So this slide really is an illustration that there are windows of opportunity. And so at birth is in particular a window of opportunity where exclusivity has demonstrated importance both in the microscopic changes that occur in the development of proper colonization, but also in terms of long-term disease outcomes related to cancer and immunity and disease prevention. So really what this slide illustrates is how narrow the spectrum is of the bacterial colonization, how bifidobacteria really does comprise the primary microbial colonization of the newborn's gut, and how specific and sensitive that newborn epithelial cell is to different effects, different antigens, different conditions that might arise in changes in the baby's immune system. And then finally, this is a recent study that really is exciting. These researchers followed 9 babies from ages 2 weeks to 14 months, and they collected and analyzed the stools to try to look at this microbial environment of their gut. And what they found is that there were big differences in what's illustrated here in pink in the exclusively breastfed baby versus any of the other categories, the exclusive breastfed babies that were then given supplements, the non-exclusive breastfeeding babies, and those that, again, had wean. And so the microbial organisms are very narrow in their scope. So the diversity, the phylogenetic diversity is very narrow, the number of species is very narrow. And it's important because what these authors suggest is that these microbial communities that are very narrow in scope seem more ready at the time of the introduction of solid food at 6 months. And in fact, they presumed that the microbial organisms that were present in the non-exclusively breastfed babies at the time of the introduction of solid foods could contribute to more disease patterns, more stomach aches, and even presumably more colic. And so it was interesting that the timing and the fact that solid foods were introduced in an exclusive breastfeeding population compared to the non-exclusive breastfeeding population in their studies show dramatic differences in the way in which those solid foods are tolerated. So oral tolerance is important. The gut colonization is critical to development of the proper environment, and also important in terms of how that baby is fed throughout the first year of life, and that these microbial organisms actually participate really actively in the development of the baby's immune system structurally, spatially, and again, temporally over time. So why do babies get supplemented? You know, why is it so common then that babies do get supplemental formula feeds? And many times babies are given supplements because of a presumed medical indication, but really what are these medical indications? What does the Joint Commission define as the reasons to not formula feed? Is it weight loss? Do you have a threshold for weight loss? Is it jaundice, hypoglycemia, or others? And we're going to touch on this a lot more in our next webinar when we talk about how to support exclusive breastfeeding. We know also that supplementation in the early couple of days in hospital leads to weaning at 6 weeks, and 6 weeks is the time frame because this is when the Pregnancy Risk Assessment Monitoring System survey is done. This is PRAM's data from New Jersey, and it's clear here that babies that are exclusively breastfed in the hospital have a 6 times higher likelihood of being exclusively breastfed at the time of that PRAM survey. So why else are babies supplemented? Well, certainly not upholding the WHO code of marketing, having formula just out and about and having it marketed direct to consumer as well as marketed throughout the hospitals definitely influences increasing rates of supplementation. And that's clear over the years that beginning in the 80s when direct-to-consumer marketing first occurred, there was this big gap between overall breastfeeding and exclusive breastfeeding. Much of how we learned about breastfeeding didn't include information. Some of it wasn't known yet in terms of the importance of exclusive breastfeeding. And of course, as physicians, we always worry. We worry that exclusive breastfeeding doesn't work, that it can't work, that babies get too dehydrated and too jaundiced and become hypoglycemic. And of course, everybody's worried about litigation, and so we have to figure out ways to really push those questions harder. What is the normal hydration status? What's normal weight loss? And is weight loss even an indication of hydration status in the first couple of days? So what are some of the reasons to supplement? Certainly if a baby has unresponsive hypoglycemia, and the supplementation should be first choice, mother's expressed milk. Second choice would be donor milk. But really the third choice would be infant formula. If moms have severe illness, if the mom's just not available because the baby's been transferred or the mother's been transferred, we know that classic galactosemia is a contraindication for breastfeeding. It is the only contraindication. If the baby's just not able to feed at the breast and mothers can't express their own milk, again, donor milk would be a preferable option. There's a very short list of medications that are contraindicated. Low birth weight, only when sufficient milk is not available. If there's some reason why mothers don't have lactogenesis, things like the retained placenta or adrenal crisis. And certainly if a mom has intolerable pain and can't express milk. But these are a lot of reasons why supplements are used and really shouldn't be used, and many of these are discussed in the Academy of Breastfeeding Medicine policy on supplementation. Another reason not to supplement certainly is that the exclusive breastfeeding rates are being monitored by the Joint Commission. And for a very long time, in the blue bar here, you can see formula supplementation in the first 2 days. We were stagnant, but what's really nice is that in recent years from our CDC NIS data, we've started to see ever slightly, but still very encouraging, a decline in the rate of formula supplementation in the first 2 days of life. So that is encouraging. And that correlates with an increase, again, for the first time, we're starting to see those lines go up, exclusive breastfeeding through 3 months and exclusive breastfeeding through 6 months, a small but significant increase in the rate. So now exclusive breastfeeding is up to almost 19% at 6 months. So it's encouraging, but we're definitely not there yet. And I think we cannot have this conversation without drawing attention to the issue of equity and disparities. And as you can see here, the purple bar consistently is the lowest bar in all the data sets that we look in terms of breastfeeding rates, and particularly around exclusive breastfeeding. When we look at 3 months and 6 months, the lowest rates are still among our non-Hispanic populations. So exclusive breastfeeding, we know, provides optimal health outcomes. It's so important to understand the science. The science is complex. No one really understands it 100% because much of it is still being worked out. And the exciting thing is that the NIH is now excited in learning more and funding more labs to investigate the human microbiome. And we know that exclusive breastfeeding requires support. And so just understanding the science is the first step, and understanding the rationale for why exclusive breastfeeding is really the first step. As I said, next time we're going to talk more about how to support exclusive breastfeeding and really what kinds of things should go into your supplementation policy. It's really how to write a supplementation policy. So that's where you really roll up your sleeves and challenge the issues. So I will stop here and ask if there are any questions, and end with this quote from Albert Einstein. I don't see anything in the chat box, and you're all very quiet. So we can unmute lines. You can certainly raise your hand. And Laura, are you still on?
Yes, I'm here.
Okay, great. Did you get anything in your chat box? Because
No.
anything. Okay, great. Well, as I said, I think it's important that to the extent we can, we understand the science and why exclusivity is so important. I'm just going to sort of pose a question to the group. Is there anything that I said that you feel you'd like to challenge, that you just don't believe it, seems just too far-fetched? Anybody want to raise a hand or allow us to unmute you or chat something into the chat box? Looks like there's a lot of call-in users, so we could certainly unmute the lines and see what people say. Can you do that from your end, Laura?
Sure. Yeah, I'll go ahead and unmute people. Okay, so everyone your line should be unmuted now if you were just a call-in user.
Does anybody have any challenges to what I presented? You're all 100% confident that now you understand and believe in the rationale to not give a supplementation.
Oh, and I have a question down here from the chat that just came in.
Okay, great.
So it says, it's from Rebecca Hambrick, and it says, if you only have 5 to 10 minutes to teach a mom, what is a quick way to teach only exclusive breastfeeding in simple terms?
Yeah, so I love to begin with where the mom's at and what she knows. So you could make a really quick, simple comment such as, I would like to teach you about why it's important to give only your milk or what the risks of formula are, but first I need to understand what are your goals and what do you understand about the importance of exclusive breastfeeding or the risks of formula feeding. So starting where the mom is sometimes is the best way to really hit on the issues that the mom has. Because if you start to, the purpose of today's talk is not to figure out a way to rehash what I just said in a way that's going to be understandable for the general public, because it just won't be. It's kind of complex, it's detailed, and it's unnecessary, really, to get down to that level. Really, the reason to understand what I just talked about today is to convince you and really to convince all of us that this issue, is based in science. It's based in complicated science, basic science. It's not all worked out. We have to keep asking the important questions. We have to keep encouraging young investigators to dedicate their careers to studying the science, because the more we know, the more we can be empowered to support moms to exclusively breastfeed. I think we have to believe in nature that wouldn't be here because we're here, many more years than infant formula has been developed, so we wouldn't be here if exclusive breastfeeding didn't work. But I think understanding why and the importance of why and really how complex the newborn's immune system development really is, first is humbling, but also gives us some confidence that we can challenge conditions that would otherwise beget supplementation, things like weight loss or things like what we used to call hypoglycemia, but now we realize is just a normal blood sugar for a newborn baby. So I think it puts us in a better position. It's the dialogue that we need to have, but not really, I don't think it's a message that should then be reinvented for mothers, for the consumer at large. But I could be wrong in that. It may be that this is what moms need to hear and need to know and need to understand. They certainly get some of these messages repackaged from the formula companies that are trying to, sell their products as being somehow a good product to support the immune system of their baby. I'm sure you've seen all the ads. So they've taken some of the science and changed it in a way that supports the use of their formula. So maybe we do need to have that discussion.
And we have a clarification question too, Lori, from Kathy Hoffman. And she wrote, are we defining exclusive breastfed as from the breast only, mom's milk only, or breast milk only, e.g. including donor milk?
That's a great question, and thank you for that question. It depends on who's asking, okay? So from the perspective of the Joint Commission, and so they're asking for a very important reason. They want to uphold best practice. They're defining exclusive breast milk feeding, and that breast milk could be mother's own milk or it could be donor milk. So from the perspective of the Joint Commission, they're really lumping together any human milk as being counted in the numerator. For the purpose of the talk today, really I wanted to make the point that it's not just about the properties of mother's own milk, and I think that's key. I mean, I think that the next best choice is donor milk, but it's not the same. The factors that, and it gets complicated, the immune properties in mother's milk are different than those from donor milk. So that mother's own milk, I said, actually contains within it not only bacteria but also immune cells, and some of those immune cells are macrophages, and those macrophages are actually activated to do immune functions. And to do those immune functions, those macrophages actually have antigens, HLA antigens, that are family specific. So the genetics of the baby, because they're similar to the genetics of the mother, recognize those macrophages from mother's own fresh milk and actually function in the baby's gut and then throughout the baby's body. And that's the best case scenario, is mother's own milk. And at the breast has importance at least for the self-regulation related to obesity. I think there may be other health outcomes that we haven't even looked at, why breastfeeding at the breast is important. Certainly when we think about breathing and sleep and maybe, the processes of sleeping and breathing and suckling, there may be differences in babies who are breastfeeding in SIDS prevention compared to babies who are breast milk feeding in a bottle. In terms of SIDS prevention, we don't have adequate data to, make any certain statements related to that, but I think that those are areas for future potentially compelling research. But I think there's a big difference in breastfeeding at the breast mother's milk versus breast milk feeding mother's milk versus donor milk. But in terms of order of preference, I would go exactly in that order. of course, you know, that order doesn't even take into account breastfeeding at the breast of the genetically non-mother, so somebody who would be wet nursing. And again, you have the benefits of breastfeeding at the breast, but then you lose sort of the genetic similarities between those macrophages. And again, it gets complicated. And nobody really knows for sure the implications of having non-mother's milk be the source of nutrition for the baby, and, you know, again, how that functions in terms of establishing the normal immune functions of that developing baby. So that's a fun question to answer because there's no right answer. I think it's really complicated. It's a good one to ask.
So, Lori, there's one final question, and did you want to have this be the last question and then wrap up the webinar for everyone?
Sure.
Okay. So Karen Womack writes, have there been any studies comparing the biomes of babies whose mothers are themselves highly allergic with those whose mothers are not?
Right, so looking at so those studies are actually, that's what's just been funded by the NIH. There's a huge study that's going on looking at pregnant women's microbiome, and they're stratifying by all sorts of disease factors, including allergies and atopy. So we don't know yet. I think that you could presume that there would be differences in the mother's microbiome. And as we always say with studying atopy and allergy, there's what we call the allergic march. It sort of goes from generation to generation. So it's not just the exposure of that index baby, but it's really what was the exposure of several generations past, and that's why we think we're seeing an increase in allergy, and it may take a few generations in the future of exclusive breastfeeding to reverse the cycle of having proper colonization and proper potential development of non-hyper responsiveness to antigens or hyperallergy responses. So, there's presumably differences. I think that the answers will lie in those centers in the U.S. now. They're being funded to look at that. But I think it's exciting that folks are actually looking.
Great. Well, this was really interesting. I know great background information for the next, kind of leading up to the next webinar you're going to host in June. So I think we'll wrap. Are you ready to wrap it up, Lori?
I am.
Great.
Thank you so much
Okay.
for everyone.
Well, thank you so much, and thank you everyone for joining us today. If you have any questions for me, you can email me at laura.burnham@bmc.org. And a recording of this webinar will be posted online along with PDFs of the slides. So I hope everyone has a good day, and we'll see you next month.